change my mind about Cassava over the weekend? What? You know what? You're right. Drug works. I did do some really interesting chemistry thinking, which I'll put in my report. There's an interesting thing about chemistry, and I wonder if there's any chemist here. It's not clear that it crosses the blood-brain barrier. That is definitely true. There's actually a lot of confirmers. But okay, this is good. So it's very hard for a molecule like this to do anything on a PPI basis. So filamin is not an enzyme, right? It's also not a receptor. So as this turns around, I want you to think about this. So this is all lipophilic right here, this 6-membered ring. And the only thing that this 6-membered ring can do is create a lipophilic interaction with another lipophilic pocket. So it can space fill. It can do that fairly decently. Like this would be a fine drug for the right enzyme, right? It fills a pocket. So now we have an oxygen over here. Here's the maybe a better representation as well in 2D. Then we have another methyl group here that'll space fill. And depending on the 3D orientation, like you can see that the conformer here is fairly three-dimensional. It's not what it looks like on paper. Fine. You have to look at filamin next and see where does this thing fit in filamin? Is the kind of small molecule that would fit really well in a binding pocket of an enzyme. It still is a little light on energetics. Like there's not really many hydrogen bonds that it can make, right? So you can get pi stacking with the benzene, but it's very hard to get much more than that. So with this oxygen, you can also sort of hydrogen bond accept. But there's very little going on here in terms of like molecular energetics. So hydrogen bond acceptors 3, hydrogen bond donors 1. Now you don't want that many hydrogen bond donors, and you don't want too many hydrogen bond acceptors. But if you have 0, right? You can agree with me that just a fat molecule is not going to do anything, right? So you get very little going on here. And again, these small molecules work really well for molecules that have a docking like pocket they have to bind into. You want to fill a pocket to stop an enzyme from working. All right, now let's look at filamin. And hopefully we can actually find a crystal structure. Here's the crystal structure, but it's only some domains of filament. So it's not the full filament protein. This is just like a very small part of it. I wonder if we can find a bigger one or make one with AlphaFold maybe. I wonder if there's like the most complete filament we can find here. Maybe we can sort by more to less residues. No, you cannot block. I need filament in the title. Otherwise this is a disaster. Does this even have anything in the title? It's a little painful to use. Give me a minute. Yeah, I don't think too many people would attempt the entire crystal structure of every domain of filament. It's kind of a beast protein. I don't think you could even do it, to be honest. It's too big. Where there's a will, there's a way, and I can probably pull these into some chemistry software. Should we try AlphaFold? I've never even tried it. So this is where filamin does its action, the so-called actin binding domain. This is mostly where filament works. This is 270 amino acids, which is like 1/10 of filamin, but this is 1/10 of filamin. So little semiphilam has to find a way in here to do something. Now the problem is semiphilam's too small. Look at this sulfate ion. It's barely bigger than this. This is just normal ions and solutes. So where do you make the hydrogen bond? Could be anywhere, right? But how many hydrogen bonds can you make? And you were trying to stop the interaction of two proteins. Very hard to do. You generally need a really big molecule for that, like 2,000 molecular weight. So 10 times bigger than semiphilam. Because you kind of have to wrap it around the protein-protein binding interface. Let me see if I can make this more clear. Like here you see a benzene coming out of a histamine, right? Here's another one, or phenylalanine. So like, could you pi stack on here? Yes. We have one benzene here, and theoretically we can make like a pi bonding interaction there, like say right here. But how will this stop nicotinic receptor alpha 7, which was pretty much useless, doesn't do anything in Alzheimer's? How would that stop that protein from binding to this massive protein? So you're looking at a 2,000 amino acid residue protein and a 200 Dalton molecular weight small molecule stopping the binding between these two. It's really like thinking about, I don't know, like a huge magnet picking up an automobile, maybe in a junkyard, right? Here's an automobile. And then having like a stick of gum here and saying that stick of gum is going to stop these two. It's going to stop the interaction. You really have to wrap around the entire binding site. And a small molecule really can't do that in general. That's why we have antibodies. But antibodies don't get into cells. So this is kind of a ridiculous premise to begin with, but I spent a lot of time on this premise just because when you look at medicines, it really makes sense to like actually give credit to the other side. Now remember, this is 1/20th of what filamin actually looks like. It's really big protein. So I'm going to stop this really big protein from binding to this other really big protein. Okay, here's this gigantic protein and nicotinic ion channel receptor. Ion channel 476 pentameric transmembrane protein. Somehow filamin is binding to this, which by the way, I don't believe, but assume it is. And little cemiflam is going to stop the binding of this guy and this guy. So somewhere there's a binding interaction. And again, this really, really small molecule, which I can add to the PDB file. This is the guy that's going to end it all. Seems unlikely. And it's unlikely because it's as unlikely as you can imagine, because why would it be likely? Most large molecules, this is where 4090 would help, right? Most large molecules, most protein-protein interactions, when you try to design a PPI inhibitor, you are not, you're going to do it with a larger molecule. This molecule is way too small. Pretty pictures, but not too useful for our benefit. So not PPI proton pump, but protein-protein interaction. Different PPI. This is medicinal chemistry. So again, I would challenge Cassava to say, where are we stopping this interaction? And they didn't do the science for that. And how do we know that? Well, let's look at their patent. Remy got a patent. That's kind of funny. As a CEO, you don't really want to be in the patent business. Let your scientists do that, you know? Just kind of cringe. Oh, was this guy scamming as early as 2012? I'm just trying to look at the date of this application. Okay, this was filed 2020, so these patents kind of represent probably the most critical data. Fake Western blots. What is this supposed to be? Percent stimulation, calcium influx of the ion channel. Okay, these all look the same. These like all could be done. There's no causality between A-beta-42 and alpha-7. They're really like just not related, and that's been proven over and over again. Okay, so what's this supposed to be? 14B. There's a label for this somewhere, but so ICV is intracerebral ventricular, A-beta-42 versus with drug. There's sham and sham. This kind of data looks interesting, assuming it's not fake. This looks like measurement error. Yeah, I'm gonna have to go through these patents really carefully. This is basically where all the science exists, because the publications don't do anything. Patents, you have to be a little bit more explicit. Well, the problem is and I'm gonna write about this, when you're trying to develop an assay, like typically none of this stuff really makes a difference. Okay, here we're starting to get a little, this is kind of what I was gonna talk about. You need stuff like this. So I'm not sure what this is AB42 binding to what exactly, but in general, what you're trying to prove is that in the presence of your drug, you're like shifting the binding characteristics of some canonical ligand. And you could do that here. I'm not sure that they ever tried. I wish there was somebody who understood the Cassava science that could actually walk us through some of this. I mean, I understand it, but the bears are going to say, oh, well, Shkreli, you're just coloring it negatively. But here's the peptide that they say it binds to. So this is valine. Yeah, let's just pull this up. I think I can create this in some chemistry software. AB42 is amyloid beta, 42 peptides, yeah, or 42 amino acids. So here's like GenScript has a little online tool. So let's do VAGCL. So it's valine, alanine, alanine, glycine, leucine. I don't think, yeah, this is like too sophisticated for kind of what I'm looking for. And we can actually look at this isn't hard to actually look at the binding. Okay, here's one from Tulane called 𝗕𝗢𝗄. Okay, so here's vagal. I can get some like chemist buddies here to talk about this, but okay, here they say that vagal and 𝗚𝗔𝗏 have this exquisitely good binding. Okay, so here's 𝗚𝗏. Okay, so you tell me where this happens. So if I take this guy, and let me see if I can rotate it. Yeah, it's a little tricky, but maybe you can get some energetics here, okay, and then maybe, so this is a bond. But the problem with this bond is we know the strength of hydrogen bonds, right? We actually know the strength of these hydrogen bonds. Do you guys know what they are? Kilocals, right? They're measured in kilocals. So yeah, I don't see how you could really get a serious binder here. You have to add up, yeah, so vagal is the binding site, theoretically, of filamin A. And we can actually do the ligand docking online. There's this thing, there's a couple of these things. So there's SwissDoc, and we could do SMILES here, and then we could do PDB, or we could prepare our own PDB. And after we start docking, we can get the docking experiment. I actually had a thing called drug-like, which I could also use. Yeah, so I can roughly calculate the binding affinity, but it's going to be fairly low. I mean, it's kilocal per mole, right? We don't have commodity prices yet now. I don't see how you could get to a level that would be reasonable or specific. Like, this would bind any peptide with low affinity. I think there's no chance at all this drug could possibly work. I think that Matt, I mean, look, he's very smart and successful in his own field. This is my field. Nobody looks at data other than control trials. You do not do a trial for fun. It's really expensive. You do a trial hoping you prove something. When you don't prove something, you really have to start over. I would do another 6-month study. I wouldn't do a phase 3 study based on this data. That's kind of an act of desperation, I think. Why is stock up today or tomorrow? Listen, when I shorted Vital, the stock went up. I was thrilled to short more. Stock went from 20 to 2, and they had to change their name. Cassava won't be Cassava next year. Cassava will be merged with some reverse merger company that wants to go public. Yeah, there's lots of exceptions to the logical and rational. How do you time the short? I don't know. Like, I just like shorting common, honestly. We're on YouTube and Twitch at the moment. I think the people who are new to analyzing clinical trials for a living, and I honestly think Matt is. I don't want to insult the guy, but new dogs can pick up the game very quickly. But I've studied, I don't know how many clinical trials. I've paid for many clinical trials. I've written protocols for clinical trials. This is a failed clinical trial. And this is a drug that doesn't make sense. There's no mechanism here that's plausible. I know a lot about proteins. If you know the binding site of the drug to what amino acid on filamin, is it a covalent bond? It's just stupid. It just doesn't make sense. By the way, nobody else has talked about filamin being a target for Alzheimer's. I mean, it could be that they're the first, but it's not it doesn't make sense. I mean, so the only person who knows anything about science, Remy is not a scientist, by the way, at the whole company got arrested. Apparently faked all this data as going to C12. Well, actually, that it is a good question. Like, let's look at filamin. We just kind of say, okay, it's a scaffold protein, who cares? But let's actually study it. Let's see, identified in 1977 from the chicken gizzard. You know what the gizzard is, right? It's an initial PNAS paper by different Wang. It's a funny name, filamin, by the way, because you have that like filament idea. Yeah, I just don't think this filament hypothesis is just hand waving. It's just nonsense. There's no such thing. It's not relevant. For example, A beta 42 binds a million things. Every one of these papers, let me see if I get a better Oh, there's an 𝗚𝗂𝘆 paper. Every one of these papers is the same as 𝗕𝗢𝗡𝗏. Just hope and prayer that my idea works. And when you tried it in clinical trials, it didn't work. There's plenty of other, by the way, Alzheimer's drugs that did have good phase 2s. Not all of them had good phase 3s. But here you're trying to take a bad phase 2 and tell me it's going to be a good phase 3. Come on. Now I'm going to get involved here. I'm going to call the FDA. No, wait, they already did that. Sometimes in science, you're just making up mechanisms because you didn't design the drug. He didn't take a filamin A crystal structure and say, okay, I'm going to hit residue 492. The valine, has a hydrophobic residue here. I'm going to put a methyl group here. It's not. He didn't do that. Where's the medchem paper? Like, where's the discovery of 𝗕𝗢𝗡 targeting filamin A? Like, structure activity response, logical drug discovery. You do screening and then you do medchem. I'm going to write down all of the things amyloid beta is associated with. How many do you think that's going to be? Tell me how many you think that's going to be. It's going to be a lot. So the idea that one of these is the true answer and everybody else is wrong and this guy's right, the guy going to jail, the company with the failed phase 2, the 150 mechanisms of actions of A beta, this is the right one, not all those other ones. What are the odds that all those are true? Insiders are holding so long. I don't know. Some of them may be getting booked. I got advice. Learn the rap game. You want to get along with guys in prison? Listen to some rap music. I'm not talking Post Malone either. Go to World Star. You want to understand rap? You got to listen to World Star stuff. I don't see Remy doing that, but you got to learn all these rappers, their histories. You ain't gonna be worried about PubMed studies. Young Thug, the whole Young Thug, Rich Homie Quan drama. You gotta know every piece of that. There's a lot on 𝗡𝗏7. I have to admit, there's like 2 papers. I'm not even looking at 2 papers show up in 𝗡𝗏7. So I'll give credit. I always give credit where credit's due. You're not gonna catch me lying. That is, there's a couple papers here on 𝗡𝗏7 already, and I just opened it. So, Cassava borrow rate, I wouldn't even worry about it. You might pay 37% for 2-3 months, but it'll spike to 200 or 300 when I'm done with it. Let me tell you something. This isn't a game. You're bringing back the old Shkreli. You guys are clowns, man. I was shorting biotechs before you were born. You're gonna get me real mad. I'm gonna raise my voice. You do not want that. I'm a reformed individual. Yeah, I was a product of my environment. So what? Yeah, I didn't make every trade right. Oroxagen was the one trade that did not go well for me. But I paid the price for it. This is what it is. The company went bankrupt, by the way. So think on that before you bring that one up. Oh, remember the metal hypothesis with prana? That was funny. People thought it was going to be heavy metals and copper that caused Alzheimer's. 𝗬𝗏𝗃 inhibitors. So actually, if you think about that, it doesn't really make sense that A beta monomer, right? You're talking about an A beta monomer binding filamin. Is that what we're talking about? Because most of the time, A beta is not in monomeric form. This is just sloppy science, man. Talking about monomers? Monomers? I'm supposed to be a polymer, and you're talking about monomers? We're making ultrastructures, filaments. You're talking about monomers. We got fibrils. If I'm going to have 70 million in one stock, I better be a billionaire. And that's a small position, right? That's 7% of my wealth. All good. I made a mistake. We're going to recover, team. We got this. We're never going to do biotech again. Let's fall back. Play my position. I didn't come correct when I saw Shkreli. That was a mistake. We're never going to do that again. But to YOLO 70 in your first biotech, get some experience. Like trade 5 million, 1 million, some other biotechs, kind of learn the ropes. You're just jumping in with the BSD, if you guys know what that is. You got the BSD out. This is the BSD. So I got 70 on X, 70 on red. What? There's top hedge funds that aren't putting 70 on a clinical trial. I have some regulatory questions about shorting, so I don't know. I was chill with him. I was not trying to Yeah, I will do a second call with him. I'm just going to triple down on the research, do a little of my looking into things. Because if the hypothesis is around A beta binding filament, okay, where is it binding? How strong is it binding? Can we replicate it? Is it a monomer? Is it a dimer? Is it a protofibril? Is it a fibril? Is it a filament? I can commission that study at about 10 grand. All right? I'll do my own study. Shkreli Labs, what's up? You call up like top Alzheimer's scientists. Yeah, how may I help you? Have you heard about the research in filamin A and Alzheimer's? Well, filamin what? Kid, stop pranking me. That's what's going to happen. There are clinical results. It didn't work. Here's the protocol. I found it. Who ever heard of a 33-page protocol? Chad, you ever heard of that? A 33-page clinical trial protocol? Man, the introduction, 33 pages. No details whatsoever. 1 month PK. Yeah, that's a good idea. $3 million. What's the results? No outcomes available. Word? It's not necessarily a red flag if a company rebrands itself, no. I have to get this with my academic login. They didn't get caught in one little Western blot lie. 4-hour half-life. Go back to the drawing board. It's not a drug. 4 hours. Can you get steady state levels in plasma at 4 hours? Let's see, 4, 1 half-life, you're down to 50%. Another half-life, you're down to 25, now you're at 8. Another half-life, 12 and a half. Another half-life, 6.25. You have 6 half-lives, right? Basically. So you have 0.5 to the 6th power. 1% of drug left in bloodstream. Or depending on how you look at it, could be 3% left. No, I don't know what ITPM is. Sorry about that. There's no proof it even gets in the brain. Wait. CSF draw. They had a CSF draw. Filamin's native shape. Altered conformation. They got Cmax for the blood. Why don't they have fucking PK for the brain? So there's less CSF than in plasma with huge variability. 4-hour half-life. Nobody's gonna like subgroup data. I'm zooming in. Let me get you that Cmax, by the way. That's gonna be helpful in the future. Maximum concentration, 1100 nanograms per milliliter. 0.6 partitions to the brain, allegedly. Right? Half-life, 4 hours, allegedly. So, 1100 times 0.5 to 6. So there's 17 nanograms per milliliter. What is the volume of blood in the brain? It's not even a liter. Half a liter, maybe. Oh, even less. 0.1 liters. 100 ml. This is plasma Cmax, yeah. But I'm taking the ratio, this 0.6 fake ratio, and saying, okay, so there's Cmax of 600 nanograms. So we're going to get 10 nanograms per milliliter. There's 100 milliliters. So we're getting a microgram of drug in the brain, a steady state drug in the brain. And they say it's picomolar affinity. Oh yeah, you're right. The drug is taken twice a day. Well, even with the twice a day, that's what, well, I guess you're right. We'll do that calculation later. It's going to be very intense. There were 2 CSF draws. Did they do different doses? A beta increased. That's what you want in Alzheimer's, I believe. Apparently all these things have errors. You read some of the errors earlier. And some of them are inexcusable, like clearly fake. I don't know.